Thank you very much for the introduction. Thank you very much for the invitation. It's really an honor to be here with such an amazing audience and for such an amazing symposium. So, well, I guess the introduction already gave you a spoiler of what I'm going to talk about. But just imagine for a moment that I give you a pencil and a piece of paper, and I ask you, regardless of your background, to draw a cell. Most likely, the first thing that you would do is draw a circle. And then, depending on your background, you will put something inside. Now, what I'm really interested in is that circle. And the reason I'm interested in is because that circle separates what's inside from what's outside and gives a cell its own identity. So this is the question, how that boundary, how that circle could have formed, could have emerged on early Earth. And the way we are approaching this is because my background is chemistry, from a chemistry point of view, and we are trying to understand what kind of molecules could have been available on early Earth and formed a first example of primitive cell, and then how that primitive cell would have evolved into the kind of modern cells that we have nowadays. Now, I am sorry to say that I'm not going to spend a lot of time talking about lipids today, but a big part of my lab is interested in the emergence and evolution of primitive lipids. And one of the key reasons why we're interested in that is because if we look at modern cells and modern membranes, you might know that the diversity of molecules that form modern membranes is just incredible. There's hundreds of different lipids present in modern membranes. There's proteins in there. There's other molecules in there. And this is something that in no way we are mirroring when we are studying primitive cells, where we tend to use one or maybe a small combination of few lipids to build primitive membranes. And as long as they form a bubble, we are happy with that. So what we are really trying to understand in my group, at least with a big part of my group, is how you go from such simple bubbles, soap bubbles, to the primitive membranes, to the modern membranes that we have nowadays. And so the way we are approaching this problem, and I have just one slide on this actually, is first of all by expanding the diversity of lipids that could have been available on early Earth. And this is just one example of a paper that we published with my first PhD student, Maya, the probably most brave student that I could ask for because she decided to join a completely empty lab. And what we were doing here was basically taking very simple lipids and trying to play with the head group of these lipids. And the head group of these lipids is important because it's what gives, to some extent, the lipid its functionality within a membrane. So what we were trying to do is to demonstrate that perhaps on early Earth there aren't just one or two different types of lipids, but there's a whole variety of them that we could draw from when we want to look at primitive membranes. And the second aspect we are interested in is I just said that there's a whole diversity of different lipids in modern membranes, and yet there are some lipids that are absolutely not present in modern membranes that instead would have formed very likely on early Earth. And so something that we are also interested in is understanding how you select for the right type of lipids, the right type of components for membranes that we find nowadays in modern cells. Now, I said that this is the only slide I'm going to show you about lipids, because there's another type of compartments that are quite present in modern membranes and in modern cells, and that in the past decade or so have been quite explored in terms of proposing models of primitive cells. And this would be called biomolecular condensates or coacervates, and this is the story I'm going to tell you today, whether primitive versions of these biomolecular condensates that are sort of the organizers of modern biochemistry, because a lot of reactions happen in them, inside the cells, whether these biomolecular condensates would have been available on early Earth. But to do this, we need to take a step back. also in time, because this is an evolutionary timeline along the lines of something that you have already seen today that was proposed about 20 years ago by Jerry Joyce. And you can see that it connects the formation of Earth through prebiotic chemistry, the RNA world, and then life as we know it. And I would say, luckily enough, in the last decades, a more holistic approach has emerged. And so we are a bit less focused on a pure RNA world and starting to understand what's the advantage of having more than just one molecule in there. And so this is just a representation of an evolutionary timeline that we've drawn. And I want to, it's a very simplified version, but I want to draw your attention on two different aspects. The first one is the peptide RNA world, or RNA peptide world. This is something that I know we have already mentioned today, and I know is very close to the research that Thomas is doing. And the reason I want to highlight this is because in the last decades, we have really started to understand that there are advantages in combining peptides and RNA, not only, for example, for the origin of translation and to understand how the first coded peptide could have formed, but also when it comes to RNA chemistry and RNA replication, and there are some examples that show that the presence of peptides could have helped the replication of genetic material. The second aspect I want to draw your attention on is that intentionally, on this timeline, I decided to put compartments later in the timeline. And the reason I've done that is because when you think about compartment, and we've heard this a bit today as well, we tend to think about chemistry that needs to happen, RNA chemistry, the emergence of molecules, of nucleotides, the emergence of amino acids, their polymerization, their functionality. Then we put everything in a cell. Then we put everything in a compartment. I'm not entirely sure we should look at this in this way, and I'm not entirely sure that compartments really happened later, and I hope by the end of my talk I'll convince you that this is not the case. And specifically, because we are talking about an RNA peptide world, the kind of compartments I want to focus on today, as I said, are coacervates. And the reason I want to talk about coacervates is because you can form relatively easily coacervates that look like this under the microscope, so just droplets under the microscope. You can form them just by taking an RNA strand, so an RNA molecule, a positively charged peptide, you put them together, they form these beautiful droplets that derive from liquid-liquid phase separation from solution. So the issue and the reason why I put these coacervates later, after the peptide RNA world on the timeline, is because the type of model of coacervates that have been proposed so far rely either on very, very long RNA strands, usually they are ribozymes, so 50, 80 nucleotides long, which from a prebiotic point of view are, let's say, not as plausible, or at least we haven't found a way to synthesize such long sequence-specific RNA strands. And the second aspect is that usually these coacervates are made with peptides that, as I said, are usually positively charged, arginine-rich, and usually these are homopeptides. Repetition of arginine for relatively long peptides. And you can already imagine that in a prebiotic scenario where you have a lot of different amino acids, it's difficult to imagine that only one amino acid would have formed homopeptides repeating itself in relatively long peptides. So the question then becomes, how primitive could coacervates be? Is it possible that they formed with much simpler, much shorter, and less sequence-specific polymers, or were actually they a late invention in the evolutionary timeline? And if they were there, how do they influence the chemistry that is taking place on early Earth? So this is mainly the work that Nina and Karina have done in the past year. And these are the, let's say, key players that we are working with. So I've mentioned arginine. This is just an example of a peptide, one of the peptides we used. This is a tetrapeptide, so 4 arginines repeated together. It bears positive charges, so we need something negative to interact with that, and as I mentioned, we use oligonucleotides. Here I've just depicted DNA. We're going to talk about DNA and RNA in a second, but what I want to mention is that very often we tend to use, people tend to use DNA instead of RNA, also for this kind of studies, because it's easier to work with DNA and it's cheaper to work with DNA. But we know from an origin of life perspective that RNA plays a much bigger role than DNA. We have heard that DNA and RNA building blocks were likely there at the same time. So what do we know about the differences between RNA and DNA, and how does this play a role here? So just to give you an idea of what we do in the lab, we take DNA, relatively short. Here we have a DNA oligo that is made of 20 nucleotides, random sequence. And here we use R4, so this is a tetrapeptide with 4 amino acids, 4 arginines. We're going to talk about random sequences in a few slides. But basically, that's what happens. You mix the two together, and you just end up with droplets, that beautiful droplets that you can see under the microscope. You start to test them for their stability, for example, towards salt. And what happens is that when you reach a certain concentration of salt, they just disappear, they disassemble. So by looking at their stability in different conditions and with different concentrations in the presence of salt, this is what you can build. It's called the phase diagram. And the important aspect you need to take from this graph is that the whole area that is under this curve are concentrations in which you do have droplets. So this area seems already quite big if we look at that, but just to give you an idea, if instead of using DNA, we were to use a peptide that is negatively charged, for example, a glutamic acid repeat, this is what you end up with. So first thing to notice is that the area underneath this curve is much, much, much smaller, meaning that the stability of coacervates made of peptides rather than mixed DNA and peptide is very different and much lower. And the second aspect that you can notice is that this curve is actually quite symmetrical, while this one is not. And the reason why this is symmetrical the fact that this is symmetrical tells us that you need to have relatively similar concentration of a positively charged peptide and a negatively charged peptide. This is not the case with DNA. You can have way more, a much wider range of concentrations of your building blocks. You still end up with coacervates. Now, from this kind of phase diagrams, what we can obtain is a graph that looks like this. And the important aspect of this is that we were trying to understand how minimal the system can be, how much you can shorten your polymers and still have coacervates. And what we observe is that when you use a tetrapeptide, so arginine 4, the minimal length of a DNA mix sequence is 6. 5.9, so rounded to 6. So you just need 4 arginines and 6 nucleotides to form those droplets there. When you instead use a slightly longer DNA strand, 12 nucleotides, what you really need as a peptide is just a 3 arginine unit. So this was something that was interesting. We decided to expand it even more. This is a matrix. The green spots are the combinations of peptides and DNA that we varied based on their length, in which you do form coacervates. It already seems quite large. But as I was mentioning, DNA is an easy model to work with in the lab, not particularly relevant from an origin of life point of view. And so we became interested in understanding what are the differences between DNA and RNA. We found that there's not a lot of people, actually we couldn't find papers where people systematically tested the differences between RNA and DNA. And so this is what we decided to do. And I'll just show you some results that I think are quite interesting. The first one is that when you use something as small as a trimer for arginine, so a tripeptide, 3 arginines together, DNA does not form coacervates, RNA does. When you use something longer, even with a dipeptide, DNA does not form coacervates, RNA does. So RNA coacervates are more propense to form compared to DNA, or at least RNA is more propense to form coacervates with very simple peptides compared to DNA. Same kind of phase diagram I've shown you before. The purple area is where you can form coacervates with RNA. You can see that it's much bigger compared to DNA, that is the blue area. So this is already something interesting. We kept exploring the differences between these coacervates. One aspect we decided to look at is the thermal stability. Something you can see is that DNA coacervates disappear over when you increase the temperature. At the same temperature, 45 degrees, the same RNA coacervates are still there. So there's increased thermal stability. We went to look at the minimal length that you need for an arginine peptide to form these coacervates. The number is much smaller. Well, it's smaller for RNA. Minimum length of a peptide is 3 than for DNA. Minimal length of the peptide is 4. What's interesting is that we also tried a hybrid nucleic acid where we mixed DNA and RNA nucleotides. And what we saw was that the minimal length of the peptide is 3 again, which means that even just adding 1 or 2 or few RNA nucleotides to your mixed strand gives an advantage to form coacervates compared to the pure DNA system. Now, what we decided to do was to pair up with someone that knows how to do computational studies, which is something that we have no expertise for. So this is work that we've done in collaboration with Rosanna Collepardo at the University of Cambridge. And what you can see here is a cluster. So basically, when you put together RNA and peptides, what happens? It forms this kind of cluster. And the interactions between the peptides and the DNA or the RNA are of 3 types. You have electrostatic interactions, which you would have expected to be the main ones. You have hydrogen bonding, and you also have cation pi stacking interactions. What's interesting is that when you go to look at those interactions and how they differ between the DNA and the RNA system, you find that the electrostatic interactions are basically the same when you have RNA or DNA. What really matters are the hydrogen bonding interactions and the stacking interactions that are much more relevant for RNA compared to DNA. Something else that we noticed that makes a difference is that RNA is much less structured. You can see it here, or maybe there. RNA is much less structured compared to DNA. You can already see this kind of helical structure for the DNA, which you would expect. The fact that DNA is more structured makes it less prone to interact with peptides. And so what we notice is that regardless of the peptide that we use, RNA is always have more points of contacts and more interactions with arginines compared to DNA. Now, I'm getting towards the, let's say, first take-home message of my talk, and we decided to push where we go back to the lab, so new experimental data now, and we decided to push the system even further. How much minimal can our system be? And so, in a way, how much prebiotic can our system be? So, the first thing that we decided to look at was playing a bit with the sequence of DNA or RNA. What you can see here is that when you have polymers that are just made of 1 nucleotide, you don't necessarily have an advantage. So here, for example, you have just solid aggregates occurring when you have a polymer of G. When you have a mixed system, you have droplets, so you have the coacervates. The stability is much higher when you have mixed DNA sequences compared to when you have, for example, polyase and poly-Ts. So from a prebiotic point of view, it's more likely to have mixed random sequences compared to just 1 homopolymer. And the minimal length is also in favor of mixed sequences compared to 1 homopolymer. We also played a bit with the different peptides, and what we can see is that even if you start using mixed peptides that have mixed sequence and not just arginine, so going again towards something more prebiotically plausible, starting to mix a bit these peptide sequences, you still have coacervates that form with relatively short peptides and DNA strands. The last set of images that I wanted to show you about the possibility that coacervates were actually present on early Earth relates to something even more minimal. So here we have a dipeptide with an 8-mer, so just 8 nucleotides and 2 arginines. As you can see, there's nothing there. Let's make this even more prebiotically plausible. Let's not use diarginine. Let's mix up the peptide sequence. You start having coacervates when you just use an arginine glycine or an arginine glutamic acid dipeptide, so a much more random sequence compared to what you would expect in a relatively short RNA. So the first take-home message of this talk is that coacervates would have inevitably formed on early Earth in an RNA peptide world, or, as Stephen said earlier, coacervation cannot not happen on early Earth. Now, I'll spend a few minutes talking about what these coacervates can do, because I said, let's see if coacervates could have formed, and then let's try to understand if they were there, whether they have any influence on the chemistry we are interested in. And so the first thing that we were interested in was understanding what these coacervates do. So we looked at whether they recruit molecules from solution. So the first thing we did was look at whether the peptides that we are using to make these coacervates are stuck in the coacervate or whether they are free in solution. Well, they are in the coacervate. But again, what's interesting is that there is a difference between RNA and DNA. There's much more peptide inside the coacervates made of RNA compared to DNA. And hopefully by now you have understood why, thanks to our computational simulations. The other thing we've done, and we were interested in, was understanding the partitioning, the recruitment of oligonucleotides. And once again, something that you can notice is that oligonucleotides partition quite well in coacervates, regardless of whether they are made of DNA or RNA. But something that is interesting is that if you have DNA in solution, and you add it to a coacervate that is made of RNA, RNA doesn't seem to like it as much as if you have RNA in solution. So there is a bit of selection of RNA coacervates towards RNA strands rather than compared to DNA strands. So this is something interesting we found. We then decided to look at, okay, so these oligonucleotides are getting recruited inside these coacervates. How much do they stick around and what happens to them? What are the material properties of these droplets? So this is FRAP. FRAP stands for fluorescence recovery after photobleaching. So basically what you do is you take one of these droplets, you bleach the fluorescence in a point, and then you see how fast the fluorescence goes back to the point that you are observing. And this gives an indication of how viscous your droplet, your coacervate is. And so what we could see here is that, what you can see here, first of all, is the time scale we are looking at, seconds. Just to give you an idea, with much more complex biomolecular condensates, that would be in a scale of minutes to hours. So what you can understand from this is that the droplets made of this kind of prebiotic components are incredibly fluid, incredibly liquid. So it's very easy for molecules to move around in these droplets. The second thing you can notice is that we decided to look at 3 different oligonucleotides inside these coacervates that have different lengths. And what you can see is that they all behave similarly, but that the movement, the diffusion of these oligonucleotides inside the coacervates is affected by their length. As you would imagine, longer oligonucleotides diffuse less efficiently or more slowly compared to shorter oligonucleotides. Something else we looked at, again, now you understood the gist, I want to look at differences between RNA and DNA, is that the RNA coacervates show a more viscous more viscous behavior compared to DNA ones. You can imagine why. There's a much stronger interaction between the components in RNA coacervates, and so it's more difficult for molecules to move around in them. So there is a small difference between the two. We are still talking about seconds, so the difference is not as large, but there is a bit of a difference. And so the last thing we wanted to understand was whether these nucleic acids that are encapsulated, that are recruited inside these coacervates, are functional in any way. So the first thing that we decided to do was a very simple model system. We decided to take an aptamer. This is a RNA molecule that, in the presence of a specific molecule that is fluorogenic, becomes fluorescent. And so what we wanted to see was if we have this already structured RNA molecule that is fluorescent, if we put this in a coacervate, what happens? Does it lose its fluorescence? So does it open up and get destructured or not? So the answer is no, it doesn't get destructured, it maintains its structure, but you can already see that when we play with DNA or RNA coacervates, we have a slightly different behavior. Here you can see the emergence of some rod-like structures. And also when we start playing with longer DNA sequences, you can start to see that this RNA fluorescent aptamer is localizing on the surface of the coacervate rather than really going inside. Overall, what we can see is that compared to the fluorescence in solution, that is this gray bar here, you can see, and you can focus at the blue bars, you can see that more or less the fluorescence is similar, but once again, we see a difference between DNA and RNA coacervates, where you see that there's a slightly more decrease in fluorescence when you have RNA coacervates. So this is not what we wanted, because we would have liked to have RNA coacervates to be more efficient than DNA coacervates, but let's see where this takes us. So the last experiment I'm going to show you is primer extension. So this is something that has been studied a lot by Jack Szostak, and that basically tells you, that basically focuses on elongating an RNA strand that is best paired with its template. So this is the kind of reactions that we looked at. We started from a primer and that base pair with a template, and we were just interested in seeing whether the primer gets elongated over time with or without coacervates. And so I just want to apologize because the next two slides are data that we collected in the last week or so, and so they are less polished than the ones I've shown you so far. But basically, this is what we observe. So over time, when you have primer extension in solution, you would have this kind of orange line here. So over time, you are extending the length of your primer to the final length that you want. When you do the same reaction in the presence of just the peptide that is an inhibitor for this reaction, you can see that the efficiency goes down. Unfortunately, when we do the same thing with coacervates, the efficiency is also relatively low. It's not killed entirely, it's not inhibited entirely, but it is not as good as we would want that to be. This is with RNA coacervates. This is with DNA coacervates. So once again, you have a bit of a decrease, but this time in coacervates, we start to have we observe a better result compared to RNA. And so the last result that I'm showing here today is something that we got by playing a bit with more minimal coacervates. So here we have 8 nucleotides, so mixed sequence DNA. We have a tetrapeptide, so we have 4 arginines. You do this with RNA, absolutely zero efficiency. There's no primer extension at all. You do this with coacervates made of DNA, and you see that the primer extension is completely inhibited when you only have the peptide, but this time when you have the coacervate, you almost recover half of the initial efficiency. And so overall, what this is telling us is, first of all, is it possible that DNA might have had a role that we had not anticipated before, that RNA doesn't have, which is making coacervates that could be able, at least with very simple sequences, to host a chemistry that RNA coacervates cannot. And more in general, to leave you with a milder take-home message, coacervates are definitely not just bystanders, but they are actually playing a role in the chemistry that we are interested in prebiotic chemistry. So with this, I just want to show you that we have decided to revise the evolutionary timeline. I've shown you that coacervates cannot not be there on early Earth, and so we decided to put them simultaneously with the peptide RNA world, and hopefully we will now start talking about a compartmentalized RNA peptide world and explore what that can do. So as it was mentioned, we just moved from the University of Strasbourg to the University of Cambridge. Quite surprisingly, we have not broken a single glass pipette, which is amazing. This is the group. This is when we were celebrating the 2nd anniversary of the group. Thank you. So to them, it goes the main acknowledgement, but thank you very much again for the invitation and for listening.